nanoHUB-U, online instruction.
Welcome back. I'm Professor Rickus. In this lecture, we're going to be talking about the cell-device biointerface. So first, we're going to talk a little bit about the importance of this interface in biohybrid devices. Our discussion today is going to focus primarily on mammalian cells, and we'll return to bacteria and other prokaryotes later when we talk more broadly about cell-device integration. So we'll talk about the relationship of the cell-device biointerface to biocompatibility. We'll talk about how cells interact with surfaces and the effect of different surface properties on cell adhesion and cell function. We'll highlight the difference between 2D integration and 3D integration and talk about some of the differences between an in vitro device and an in vivo device. And that discussion will take us into the tissue device response for implanted devices. So, first, thinking about the importance of the cell-device interface for biohybrid devices. In previous lecture, we talked about this Ray biohybrid robot that had living cardiac myocyte cells that formed essentially an artificial muscle in this metal and polymer-based robot system that could swim via light-controlled. So in this case, as well as also imagine if we had an electrode array and we wanted to interface that array with some cell. In these cases, right, the cells must adhere, survive, and function within this artificial environment. And this really comes down to this cell-device interface as this first line of action where this adherence that affects survival and function happens. Okay, so what's the relationship? You may have heard this word biocompatibility. So here's a definition of biocompatibility that I favor. So biocompatibility we'll say something is biocompatible, a biological performance in a specific application that is judged suitable to that situation. Now that seems somewhat vague and perhaps unsatisfactory to you, but let me tell you why I like this definition. If someone tells you that something is biocompatible, blanket biocompatible, don't believe them. There's no such thing. Biocompatibility describes a two-way interaction between the host, that is our living cells that we're interested in, and the material or device, okay? And so the performance of both the cells and the material and device, what we measure as important function or performance is situation dependent, right? And there is always a two-way interaction between these things. So there is a host response. In other words, do the cells function the way we want them to when they're integrated with the device? And there's always a device response. Does the device function when integrated with the cells? So notice what's not in this definition. There's nothing about being inert or no interaction, because that almost nearly never occurs. There's almost always some interaction. And the question is it an interaction that we want and is beneficial to our biohybrid device? Okay. So let's talk about these interactions a little bit more specifically. How do mammalian cells attach to a surface? So here in the top, we have sort of a zoomed out view, thinking about the cell and the surface of our device that it's interacting with. And if we focus in on this junction area now and think about what's really happening. So here we've got to zoom in at the cell membrane. Remember, cell membranes have proteins that are embedded in these membranes that act as their receptors, and they really relay information from the outside of the cell to the inside of the cell, both through cytoskeletal components and through soluble signaling. So how does the cell really attach to the surface? Well, there are extracellular proteins, often produced by the cells themselves, that are laid down, and it's these extracellular proteins that act as adapters. They bind to receptors on the cell membrane, and they adhere and interact with the surface. So in this way, thinking a little bit more directly about the cell-device interface, we can see that the the physical, chemical, mechanical properties and biological properties of our device surface influence our cell function downstream by interacting with extracellular proteins that interact with receptors on the cell that relay that information to the intracellular part of the cell, including the cytoskeleton and other components of signaling networks that can ultimately affect gene expression and the phenotype or function of our cells. So there's two key points here. One, that the surface properties of our device influence directly the cell adhesion, survival, and function, and that the device is really directly coupled to the cell's intracellular signaling via protein interactions at the interface. So now this gives us a framework to dig in a little bit more deeply, and we can think about how do surface properties affect cell attachment. Well, one of the most basic surface properties is the hydrophobicity or hydrophilicity of the cell surface. And this will affect both directly the protein adhesion, including those proteins that cells use to attach to adhere. And we can also think about it from a more zoomed out view and how those cells how they will spread out and lay down on the surface in a hydrophilic state versus a hydrophobic state. Another very important property of the surface are the mechanical properties. One of those properties that's been looked at extensively is elasticity, the elasticity of that matrix. Essentially, you think about how stiff or how elastic that material is that the cells are attaching to. And here's some data from some now classic experiments that demonstrated that even the stem cell lineage commitment, so in other words, what type of cell a stem cell turns into, right, for these mesenchymal stem cells can be driven by mechanical properties. So these same cells in the same environment, varying only the stiffness of the substrate, you can induce these cells to differentiate into brain-like neural cells if they're on very soft materials, right? And as that material gets more stiff, then you can induce them to differentiate into muscle, or at the most stiff material, you can induce them to differentiate into bone-like cells, right? So this is, as I said, a classic example and a dramatic one that shows how the mechanical properties of the surface can dramatically influence cell fate and their function. So another property that can be very important is the topography, particularly at the nano and the micro scale. Remember, these proteins that are adhering, proteins adhere really, they're living in the nanoscale, right? And cells are micro scale structures. So here we have two materials. These are silica-based materials produced in my lab that they are identical chemically in every way. They've only been processed differently in order to change their surface topography. And so here we see an atomic force microscope image of these two materials, one that's very rough on the nanoscale, and notice these axes even are different. So this axis here is about 150 nanometers, whereas this, you can see this material is very, very smooth, sub 30 nanometer surface features on this material. And just by changing the surface topography, we can dramatically affect how neural cells attach to this surface. So on the nanoscale rough material, we see almost no neural attachment to these surfaces. While the same material of identical chemistry, now that's much smoother on this size scale, we can get very strong and nice neural cell attachment to these surfaces. So I put this figure up just to show you and sort of put in your mind the complexity of this interplay. Again, we're looking at from this surface or scaffold interacting through these protein layers with the cell versus multiple different types of receptors and adhesion molecules, coupling into both the cytoskeletal elements as well as the complex signaling network of the cell, and as well interacting with the soluble and interstitial volume in space and the signaling that's happening in here, as well as any other physical inputs such as pressure and shear force from, for example, fluid flow. So all of these things are interacting to influence how those cells adhere, survive, and function. Okay, so we've mostly been talking about cell biointerface in a two-dimensional context. But quite often we're also looking at cells in a three-dimensional context. And so I put up this cartoon here that I made as sort of articulate these two things. One, looking at the cell attaching to a planar surface versus cells that may be within a matrix, artificial, that may be integrated into a device. And you can see how they're interacting with and adhering to the artificial components can be very different. So in a two-dimensional, we typically get a very flattened cell shape and actually an abnormal distribution of these receptor adhesion ligand complexes. So there's often this forced polarity and abnormal morphology in this sort of planar orientation. And cells are very dramatically, again, you can see how their morphology even changes, again, as we vary stiffness, as we looked at before. So now you're sort of seeing the attachment points and how the cells sort of spread out and have these different morphological shapes, which you can imagine has a direct impact on the signaling and how they're functioning and what's happening internally to the cells. Whereas 3D is generally considered a bit more of a native environment. This is how cells typically live in a three-dimensional sorts of environment. And so they often have a more natural distribution of adhesive sites with the material that they're interacting with, and less of a forced polarization state that can influence cell fate. And in many ways, the sort of cell fate and function in most of the ways that you can measure is often different for the same cell if you are to interface it in a three-dimensional environment versus a 2D environment. Now, for the sake of devices, there are different types of three-dimensional materials that we often use to house cells in devices. They can be synthetic, such as a PEG-based system. It can be natural and non-native, such as alginate polymers. And they could be natural and native, such as collagen and laminin types that we would often see mammalian cells, for example, surrounded in the body and in natural organisms. Okay? So cells can actively remodel and shape their 3D environment. So again, this is that two-directional communication between cells and the materials that they're surrounded in. And here's some examples now. We put in these different three-dimensional environments. They can also not only being sort of natural or non-natural or native or non-native, we can also have materials that are either degradable or non-degradable by the cells. And you can see that in the non-degradable materials such as alginate or this artificially cross-linked hyaluronic acid, another natural component of many tissues, that in these cases the cells are fairly confined, right? And their morphology is somewhat rounded. When you put the same cells in a degradable, either collagen or in this case now a differently cross-linked hyaluronic acid, one that is degradable, you can see that the cells now are able to change their shape by degrading and manipulating the environment in which we put them in. And that's something that cells naturally do in their native environments. Okay, so we've mostly been talking about in vitro. We've talked about 2D and 3D, but let's also talk about in vivo. So in vitro, we're typically putting the cells onto or into an environment. But in vivo, we're often integrating the device into the tissue. And with that often comes injury from that integration, broken capillaries, other tissue damage, and a little bit different of a tissue response. And so because of this acute injury in this more complex tissue response, because now if we're in a whole organism, we often have an immune system and other functioning components going on. And so this complicates our cell-device biointerface. And so one of the things that typically happens when you put a large engineered device into the body is what's known as the foreign body response to the implant. And this happens over a course of time, beginning with an initial injury or implantation. And there's often initially protein adhesion and inflammatory cell infiltration from that injury. And then we have a continuation from an acute to a chronic effect, both at the material interface of our device and within the surrounding tissue. And again, this propagates from an acute inflammation into potentially chronic inflammation and ultimately an encapsulation of our device with fibrous tissue. Obviously, something that we often don't want if we want a good, healthy integration of our cells and tissue and our device. And so the end result of a device being put into the body is often this sort of encapsulation and fouling due to this fibrous capsule that's formed around here. We have an image of a sensor that's been placed into the body. And around the sensor membrane and the sensor components, we have protein adhesion and fouling of that device, as well as a fibrous encapsulation that sort of forces away the cells of interest, the natural cells and the capillaries, and typically the systems that we want to interface in. So here's what this can look like in the brain. Here's a reactive tissue response to a neural implant. So in our last lecture, we were talking about the brain-computer interface and neural implants as a type of biohybrid device. And what you see here is a very typical, after that, in this case, an electrode was removed from the brain. We can see the site where the electrode was, and you can see all this fibrous tissue and what's called a glial scar. So this scarring around. And so now if you look at some of the individual cell components, we can see the resident immune cells of the brain, the microglia, and the other glial cells, which are reactive and have been activated to form this glial scar around the cells. And now most often the cells we're interested in interfacing with for the brain-computer interface, the neurons and its axons, you can see, have now been pushed away and are no longer near the electrode surface that's depicted here. So this is obviously something we don't want quite often when we're making our biohybrid devices. We want a healthy integration of cells and tissues with our engineered components. So there are, I think, probably the two winning and probably most promising approaches to avoiding the foreign body response is kind of twofold. And one is to make our devices smaller. The smaller we make our devices, if we can make them on the size scale of cells, we have much less of an injury impact, and they're much more able to mechanically match the tissue. And that leads into our second approach, and that is to make the devices themselves less foreign to the body. And so this has approaches of both chemically mimicking the native cells as well as mechanically matching the tissue so that there's not chronic inflammation and the cells and the surrounding tissue see the interacting device more as self and don't attack it and wall it off via this scarring and fibrous encapsulation. Okay, so coming up, we're going to move into talking about cells as sensors, and then we'll move on to actuators and talk more about cell-device integration. And here we'll also bring back in our bacterial friends and the prokaryotes and talk about their integration in devices, as well as continuing our discussion on eukaryotic cells and mammalian cells, and talk a bit about bioencapsulation and how we integrate these cells into devices in a broader sense. So I hope to see you next time.